I think there's not much summary needed. The talks are really great and very inspiring. I'm sure there'll be a lot of questions. Certainly we had several talks on filaments and motors, be it actin and myosin or microtubules and kinesin. The final talk, well, that sort of morphology of tumor growth, and over the previous days we heard about rheology of tissue growth. I don't know if there's any sort of crosstalk between the two formulations. So with that, let's just open the floor. Is anyone okay? All right. That's fine. So if not, let's just start with, so we had, let's see, actually the modelers outweigh the experimentalists on this panel, which I think maybe was sort of by accident. So I guess one thing, at least as a theorist, I've been hearing over the past couple of days, which relate to this obviously, is that the modelers say, well, I build a model and I run a simulation or do a calculation, and it looks like the experiments, and then of course one tries to quantitatively compare with experiments. And I guess I'd like to hear, at least from a couple of the modelers, maybe are there predictions that one could make as opposed to retrodictions? And yes, there are. And so maybe if you could discuss sort of that in terms of maybe some predictions of your models, any modelers willing to say something, and then how they're interacting with the experimentalists to test that, sort of the modeler experimentalist interface, etc. And I guess also I'm a little bit biased. Professor Joanny, you didn't get a chance to talk about the contractile ring model, if you are so inclined. I don't want to put you on the spot, but
So you want me to talk about contractile rings?
Well, in Margaret's experiments, the myosin, so you first need this critical load in order to set this traction force so that the focal adhesions start to do their thing, and then you have this organization of the actin network, reorganization into these filaments, into bundles of filaments. Presumably the myosin is sort of acting as a bundling agent and as a pulling agent, there. So that's somewhat, it's different. The structure looks different than the contractile ring, because obviously this isotropic network, whereas the contractile ring, you have this nice structured ring. So there's sort of similar processes perhaps going on in terms of actin and myosin.
What we did about contractile ring is the very early stages of the formation of the ring. Now, it's known from many experiments that at the equator of the dividing cell, you have an increased density of lysosomes. So in the language that I was using, there is increased activity. If you have a gradient of activity, you need to have a flow. So there is a flow from the pole to the equator, and the flow couples with the orientation of the filament, and because of this coupling, it induces the ring, which means that the actin filaments are oriented. Now, I think if you look at the later stages, what you were saying, that there was some analogy with what Margaret was saying, is perfectly true. Yeah. That as long as the filaments are organized, myosins are more recruited, and it looks pretty much like what Margaret was discussing.
I think the boundary conditions are just different. I mean, it's even the same set of proteins you find there. It's not attached to the surface.
It's free.
So with regard to the theory of the contractile ring, do you also investigate the polarization vector? Because I guess the initial theory didn't worry about the orientation of the filament.
So the way we did it is we assumed that before this flow is created, the actin filaments are randomly oriented, and then they turn around the ring like this, but there is no polarization. Now, kind of intrinsically, if there were a polarization, the contractile ring would be rotating. Because then, I mean, the symmetry allows it to rotate, so it should, whatever the theory. It turns out that there are experiments by Daniel Liebling, Rockefeller, who on yeast cells sees contractile rings which are rotating. Then we would have to put in polarization, but we haven't done it.
So I have a question for Margaret and maybe Jean-Francois about that bundle formation. What's known about how so sort of what I want to say is, how do the myosins get recruited and reorganized? What's known about what tells them to go to the bundles and organize in a certain way? Is anything known about that? Is it purely self-organized? Are there other signals that tell them go and kind of arrange in this? Because they look sort of banded, right?
So you saw in like my movies, you can keep the myosins assembled and they'll become disorganized. But then they'll self-organize into that banded structure. So you don't need directed assembly locally, but in the cell usually you control the where myosins get phosphorylated, and the phosphorylation of myosins directs their assembly into mini filaments. And if they're not phosphorylated, the motor proteins are sort of curved onto themselves, so they can't actually interact with actin filaments very much. So I don't know if you noticed at the leading edge of the cells, right below the lamellipodia, you can see those myosin mini filaments appear to get incorporated into the stress fibers.
And what about in your contractile ring work? Is it purely self-organized?
No, everybody says that microtubules belong to the recruitment myosins. So you have these microtubule asters, spindles could be there, and apparently they regulate the phosphorylation of myosins.
I have a question.
Let's just ask each other questions. So the issue of filament polarity always comes up. It would be nice for both of our experiments to have a measure of what filaments are polarized in what direction. But experimentally, especially when you're trying to image live, that's nearly impossible with current techniques. And likewise, we can barely detect the aspect, what orientation of the myosin mini filaments are. So are there other kind of parameters we could measure that are accessible by the light microscope that would be a good characterization of coarsening or order of bundles?
Can't you tell by the way they move? Because the myosin goes towards the plus end. So if you can see which way the actin's moving, would you know which way
relative to the myosin?
Yeah, so the myosin's always going towards the plus end, so if it's actin filament's moving the way, then you know where the plus end is.
Yeah, so that's if you have a single filament motor interaction, that works out well. But when you have a bundle, at least in cells, you can't really distinguish. There's so many filaments. Each of those filaments is many filaments, and actually the orientation of those filaments interacting with an individual mini filament can actually be different. So, but looking at trying to distinguish at a point along that bundle the relative motion of the actin and myosin, I'm not sure if we'd be able to characterize that.
How processive are mini filaments?
How
processive? How long do they stick to actin?
Quite long. I think the half-life of the myosin is on the order of 2 minutes, so about the same lifetime of the actin filaments. So actin goes in and out very rapidly, but the myosin and actin is a very long interaction.
I'm going to be a little provocative here. So I see most of you look to me like physicists, but you're doing what looks like biology to me. Now, I understand the impact of the biological research you've done on physics, but I don't know the impact that the physics you're doing has on biology. Do the biologists care at all about what you're doing? Are your insights having any impact on the biological community, or have you become biologists and are doing things, the same kinds of things that they're doing? I've seen a lot of movies you've shown today that some were taken by biologists 20 years ago. They look really similar to your movies. Maybe you have 3 or 4 more colors, but you know what I'm saying?
More colors is a big deal.
I understand it's a big deal, but does it mean that you become biologists and you merge into them, or do you show up, if you go to a cell biology meeting, I've never been to one in my life, but maybe you guys go to them, do you show up as something different? Are you bringing something different to them, or do you just blend in? Do you know what I'm saying?
Yeah. I think biophysics, at the single molecule level, is a great example. So you have people that measure forces, look at rates, quantify rates, take images, quantify structural EM images. But at least in cell biology, just finding somebody who will quantify cytoskeletal dynamics by using, image processing algorithms is pretty rare. I mean, there's 2 or 3 groups that specialize in that. And I mean, yeah, the technology to analyze those images is difficult, but interpreting those images, I mean, so a lot of biologists say, oh yeah, a force is required, but no forces have been measured, right? So.
Yeah, but do they care?
Yeah, they do, because it's all morphology changes. So they want to know forces, right? They want to know how a cell regulates its shape or interacts with its environment to direct migration. But
so now in cell biology, you start seeing more and more papers that biology guys who aren't trained from a physics degree but came up through biology are starting to say, oh, that's important questions, and start adopting that approach. I mean, what are the measurable quantities that show that you're having an impact on the field?
I'll try to comment on that. I think that there is a move definitely in cellular biology and systems biology towards what people sometimes call quantitative biology, like trying to do more and more quantitative experiments and compare to quantitative models. I'd say that there definitely are people who don't care. I mean, there are definitely biologists who don't care at all. But there has been a kind of, I'd say, maybe the last 10 years, this big shift towards more quantitative experiments, and a lot of kind of more cutting-edge work is more quantitative. And so I think it's having, and I think that has been driven somewhat by physicists, also by biologists who realize that having more quantitative information can help them, like Margaret was saying, better understand their data. I mean, so I think it's having an impact. And I guess the way I kind of personally see it most is in larger and larger numbers of biologists who just realize that they need something more to help understand their data and are just incredibly excited about collaborating with someone who has enough math skills to do some of these models and be able to compare to what they're doing. I definitely have more people kind of propose collaborations that I can really do. And I think that, again, that's not everyone, but even biologists who don't have those quantitative skills realize that that can really help them, and they kind of want that more and more. So I'd say it's having an impact. I'm not sure how you measure that, other than my measure of discussions and what people are excited about. But yeah.
I guess I've noticed a real culture difference between physics and biology. In physics, we have a natural division between are you an experimentalist or are you a theorist, right? That is unique to physics. You don't see that in biology. In biology, they're just experimentalists by and large. And I've noticed that when we submit papers and we have some model, in physics, it's perfectly respectable to write a paper that's purely theoretical, say, I have this explanation for this experiment, and see my model fits that data, and you can publish a paper in a perfectly respectable journal. You have a lot harder time doing that in biology, because in biology it's like, well, I have an idea, I have a model for this system, and the biologists say, well, that's nice, go and do an experiment and show me that that's true. And the reason for this, I think, is twofold at least. One is because some arrogant physicists tend to come across as very arrogant to biologists, even the ones we don't think are. But the physicists who went in very early on, said, we're smart, we're physicists, we can do this, step aside, and left a very bad taste in the biologist's mouth. So theory got off to a bad start to begin with for that reason. And also because biology is a very complicated system. So, in physics, we have the spherical cow model, right? You just simplify it, and you work very hard experimentally to make a very simple system. In biology, you can't really simplify the cell too much if you want to do things in vivo. You simplify it, it dies. So, therefore, you have that problem. That this is why the biologists say, well, okay, do it in an experiment where you've got all the complication in there, and let me see that what you're saying is really the case. I think on a more
you're saying they don't care about all this in vitro stuff and these simplified models of your colleagues sitting next to you?
There's always the question with in vitro stuff as to whether it applies in vivo. And in vivo can be, I think of it as being in a cell or a tissue culture, but some people think in a tissue culture is in vitro, and you got to get the whole animal, right? That's their idea of in vivo. So there's...
No, I've talked to NIH, people, and they say, I'm studying a model system, and then they tell me, and then we have this discussion about the model system I'm speaking about, and then I began to realize that for them, a model system is like a mouse cancer where you look at the cell and the cell looks just like the human, and you look at the tumor and it looks just like the human, and you give the drug and it looks just like the human. And I'm saying, no, I'm using hard rods to model DNA. And they go, that rod's not charged. It's impossible to model it. And then I began to realize the word model is very different in biology than in physics. And just even the language was completely used differently, and at that point I kind of hung up.
The related question that was, do you go to biology conferences? I mean, I haven't been to a biology conference.
Yeah,
I mean, Margaret, I imagine, probably has been to a biology conference.
Yeah, no, ASCB is my favorite conference of the year. I mean, they have more mechanics there than APS.
How are you received?
So there was an invited session on cell mechanics 2 years ago. There was a pre-meeting session on cell mechanics. I mean, it's received. There's not enough people doing it. Not enough experimentalists. I mean, there's more, and then the number of posters there is just exploding. I mean, it is, and the people are very receptive.
Would you give the kind of talk you're giving here?
No, I would change it a little bit, but I mean, actually, no. I would say about 40% of the talk is one I gave there. So, I mean, and people, especially in the cytoskeleton field, I mean, and Paul brought this up yesterday. I mean, in the 70s, I mean, and earlier before that, people looked at cells and looked at it as a material. It wasn't until molecular cell biology came along and genetics came along and made us realize how complicated it was. So I feel like, biologists they look at these things under the microscope all the time, and they have all these interesting physical questions, and they want to know the answer to it. So as long as you're not talking to, somebody who's obsessed with genetics, I mean, most cell biologists love thinking about physics.
Yeah, in fact, I think before huge advances in genetics, there were lots of cell mechanics studies.
Yeah,
there's an old paper from like the 30s or something where Crick did magnetic cytometry with a magnet, just holding a magnet above some cells with beads embedded in them. So, and that was quantitative. And I just wanted to make the comment that, speaking of physicist arrogance, not all biologists, rather than put it this way, a lot of biologists do know math, and a lot of them are really. I mean, some of the best, say quantitative biologists I know don't want to train in physics at all. They're just biologists, and by that same token, lots of other fields of biology, like ecology. I mean, you can't write a paper in ecology without having some crazy, sophisticated model for predicting the dynamics and stuff. So I don't know. I just don't want to buy into the idea that without physicists, biologists wouldn't be quantitative.
I think it's different.
Can you articulate in which way it's different?
So I think the cell biologists, the ones that look under a microscope, have asked the really interesting questions about sort of mechanics of their systems. Because they're not wedded to this idea of linear elastic materials, they're willing to ask really crazy questions, right? And they've looked at tons and tons of cells. So I think their intuition about what questions to ask is really good. But I don't think most biologists are willing to put in an effort of spending, half their lab developing image processing algorithms to answer a question. They're more willing to draw a chimograph and say, oh, this is, a flow speed. So they're not the amount of time they're going to spend their time knocking down certain proteins and asking sort of molecular questions. And so there's a range, but I just think that the effort they're willing to put in, they want to know the answers, but they might not be willing to put half their lab on developing new methods. I mean, some are, but
just to go back to Seth's question, but I guess to go back to Seth's question, on a personal level, what I found a way to try, I care about being relevant to the biologists and doing something they care about, so I tend to work with biologists, people who are biologists, because they know what's relevant, they know what are the issues in the field, and they'll say, oh, well, if you do that, no biologist will care. Do it this way, and then it makes a difference. And also because they know what's going on in the field. So I find that a useful symbiosis.
But you wouldn't say you're a biologist.
No. Part of it's your attitude. I don't know how to explain it. Part of it's that the biologists, all right, this is an oversimplification. Physicists come in and we say, how does that work? You know, and the biologists come in and say, they like to identify and classify. And
they want to know how something works, but it's just some aspect of physiology, not a physical.
Yeah, and I think it's because partly in biology it is so hard to understand, quote, how it works. First of all, they're trying to identify what protein, what genes are involved in this process, right? And if you say, okay, well, I know this is the promoter and I need all of these to enhance transcription, then you say, well, how does it recruit the polymerase? I went to a workshop on this once, and it says, well, you need all these on the DNA before the polymerase will come down and transcribe the DNA. And I'd say, well, how does it recruit the polymerase? And they'd all kind of just look at me like, we don't know how to answer that question, so we don't even ask it, right? And it's this sort of, I think, a difference in attitude.
I'm just more biologists here.
Defend themselves. I just think it's a big field. There's lots of different kinds of biologists. I mean, there's been people asking, really quantitative how questions, Lance Taylor, Shinya Inoue. I mean, there's these people who've been, for their entire lifetime. It's just a subset of the group of biologists that most physicists would, feel would have a natural interaction. I mean, the first time I went to a biology conference, I was scared and decided I was never going to do a postdoc in a biology lab because I had such a bad experience. They just laughed at me. So I think you were there. I was so traumatized. But, I mean, then you find the biologist that it's more natural to talk to, and then you just move from there.
I think biologists are more open now, and they are much more welcoming to people coming into the field and being quantitative, much more so than we would be if you were, say, doing high-temperature superconductivity and some biologist wandered into the conference and say, oh, well, I have an idea. I mean, you wouldn't necessarily take them seriously. They're, I think, much more open. I think another move you can see is this thing called... What? Right, the biologists can't even, they don't even know what you're talking about. But I think another move you see is systems biology is coming online. And systems biology is the idea you don't work on just one protein, but a whole system, and you bring in quantitative analysis. And I think that is beginning to work its way through the system.
Can I ask you a kind of question not related directly to this physics versus biology thing? I just wondered, like everybody up here, we have people studying cells on a molecular level, and then people studying whole cells or sometimes collections of cells. But I wonder if there's if you think that I don't see the connection yet between cytoskeletal dynamics and multicellular dynamics, and I wonder, if this is the level at which you have to coarse grain in order to say treat the tissue as a fluid, but treat the individual or the subcellular stuff as an active gel, and connecting those two is just, it's like trying to connect quantum mechanics to classical mechanics. There's a barrier. I don't know.
I mean, I totally agree. I wanted to raise the same question. For example, for me it would be, so I use some characteristics of cells. So a cell has, say, diffusion coefficient or diffusion time such and such, or proliferation rate such and such. For example, for diffusion, the question is, how can you get this 5 minutes diffusion time, analyzing actin dynamics or cell matrix adhesion? Can you get these? Then, if yes, I can take this diffusion coefficient and move to a larger scale. But
isn't it something similar to saying, there are people who look at electronic properties and there are people who assume that there's a spin associated with the atom and then try to study how that affects the properties of the system. So they are connected, but at some level they are not connected.
Well, the whole motivation of aim of physics is to connect micro and macro scales to go for something.
I'm going to try to comment on this. Maybe I'll try to be provocative like Seth was. But, so, is that on? Okay. Is that working?
No.
All right, I'm just going to yell. So, physicists sort of like to pretend that we're all about unified theories and, we're going to connect material properties about different scales. But, it's actually not true at all. Like, the way physics actually works is sort of as a series of successive better and better approximations, right? Like when people started looking at electrical conductivity, they didn't start from quantum mechanics because it didn't exist. They started from the Drew model, like making the simplest model that with basic assumptions that might actually get you somewhere. And then when that fails, you sort of go on and make a better model, right? This is the way kind of a lot of models in physics actually developed in history, not from like, writing down the Schrodinger equation and then deriving like the properties of a fluid, right? We like to pretend that that's what happened, but really it's not what happened. So I'd say what's kind of happening in biology is really the same thing. You start with a problem and you figure out a model, and then when it fails, you make it better, and then that's kind of the essential iteration that just happens in physics. And that this kind of constantly having to worry about grand unified theories is really kind of a distraction from actually going and doing some interesting science. So that's a provocative thing.
I have a statement, maybe people can comment on it, but I've noticed that the biologists, one of the big things these days is signaling networks and cell signaling and all the cascades and reactions that happen. And the biologists often feel that that is the key to understanding the system, is if you can identify the signaling network. One thing that I think maybe is coming online more is not just chemical signaling, but to what extent is mechanical transduction between cells part of something that influences how the cell responds. I mean, rather than saying, okay, I have to have, a delta and a notch receptor and that sort of thing. You guys have comments on this? Okay, he agrees. But I think maybe it isn't seen as much as perhaps the role it actually plays.
There's a question from the floor.
Maybe let me ask a simple question, then bring together the microscale and the entire cell. What do you know about bringing together microtubules and actin and the crosstalk of it? So, for example, I heard that focal adhesion detach if microtubules point to it. Is there something more known on this topic?
You definitely have switching, of course, between microtubules and actin. Actin's and microtubules, as I was indicating. And I believe that Steve Gross once told me that in order, it's a motor to switch from going, or a cargo to go from the microtubule to the actin, it actually has to start going towards the minus end, towards backwards along the microtubule, and then it'll switch on to the actin. So, there's some sort of coordination as to how these things.
And so I don't think he's asking about motors. I think he's asking about the actual cytoskeleton, right?
Yes.
How they're linked together.
I don't know. Some people know about Margaret Gardel.
Well, there's the famous or infamous model of tensegrity, right, that was developed in the 80s, that because microtubules are these very rigid rods, that they could be compressive elements in the cell, whereas the actin is mostly the tensile elements, which seems to be true to some extent. My personal bias in microtubules is if you look at them in the cell, in most cells, they tend to be very dynamic in a migratory cell, so they sort of, my postdoc advisor likes to call them magic wands. They sort of go and they signal something locally, and they modulate rho kinase activity that affects myosin activity. So changing the dynamic instability of the microtubules can actually alter the actin cytoskeleton through chemical signaling by changing myosin activity that way as well. But you're right, the observation from Cavarina that you basically can have a microtubule grow towards a focal adhesion. That could be explained either by a mechanical cue that you have this compressive element locally that reduces some of the tension in the actin and promotes disassembly of the focal adhesion, or it could be a chemical cue that changes the rho GTPase signaling locally that then disassembles the focal adhesion. So, and intermediate filaments don't exist.
Are there proteins that connect actin and microtubules?
Yes, so,
Actin,
well,
things like this?
it's not clear what they are. They haven't really been identified. I mean, there's hypothesized to be some, but
So there are, I think, in recent times, the biologists started to just look at interacting proteins. So I think there's an exonucleated protein form, and there's been some recent papers where they write that they affect microtubules as well. So I think that's a field the biologists are now into, and I think it will take some time for physicists to start working on that. I think it starts right now more or less.
But formins can bind to microtubules and they nucleate actin, so it could potentially act like a cross-linker.
So that was just an example. So it's just the biologists do right now.
What are the roles of actin and microtubules in prokaryotic cells?
That's not.
No, there's homologs.
Yeah, homologs.
Homologs.
Yeah, so actually,
Right.
Yeah, so actin plays the role of microtubules in prokaryotes, right? So they set up the contractile ring, and then the microtubules play the role of the actin and cytoskeleton in transporting things to the end of the cell to promote cell growth. But actin undergoes dynamic instability in bacteria.
So I know not much is known about actin transport in bacteria.
Yeah, I mean, they just discovered these. I mean, they're just willing to call it a cytoskeleton now in bacteria. So,
Do you have another question from the floor?
What's known about the collagen network? You showed these pictures of it being restructured. Is it a covalently bonded network, or is it a physical network? And also, a second question is: are there examples where the collagen network is aligned or in a liquid crystalline state, and then the flow goes the other way instead of the cells restructuring the network? It's the network which
So,
the cell.
Paul might know more about this than I do.
I know that there are liquid crystalline arrays of collagen that are set up by, I think, animals that build shells around them. I don't know if that happens very bone though, humans, but I don't think that's liquid crystalline collagen. But and the idea is that you set up this liquid crystalline domain into which you can now calcify something to make this oriented thing that the collagen goes away. But in soft tissues, the collagen is first assembled, I think, non-covalently, and then it's covalently stitched together.
By the cell?
By the not necessarily the cell that makes it, but a cell that's embedded in it.
What does the cell secrete to cross-link the collagen?
There are two enzymes. One is lysyl oxidase, and the other one is tissue transglutaminase.
Okay.
And that's gotten to be a big deal in diseases like liver fibrosis, where the organ gets stiffer, and kind of similar to what Margaret talked about, the stiffening of the collagen matrix in a fibrotic organ will contribute to the dysfunction of the cell. And at least in animals, you can make the cells act more normally by preventing the covalent cross-linking the collagen network. So you don't actually destroy any proteins, you just cross-link them more lightly, and that somehow lessens the elastic modulus, and the cells revert a little bit. So I think there's some glimmer that sort of tissue biology or cancer biologists are somewhat interested in the idea that the mechanics of the outside environment does impact the way the cells behave, very related to what Margaret showed us. But there is that mix, and animals are somehow regulating that a lot, but it's really unclear what flips the switch.
Okay, so if there are no further questions.